Right. So, this is well explained in something called Jablonski diagram that I was trying to explain. So, similarly, most of the things I have explained to you. This is the electronic ground state, this is our ground state, right. From ground state we are just considering it went to S1 and S2. So, therefore, this is our absorption spectrum here, femtosecond time scale, right. From S2 it came to the lower vibrational state. From there again by non-radiative or by losing something it goes to S1, and from there again come to S1 ground state, S1 V0. From there it emits lights that called fluorescence. This happens in nanosecond. What was absorption? femtosecond. Fluorescence nanosecond. कितना slower हो गया? 10 to the power 6 times. So, 10 to the power 6 is million, right. Million times slower is our fluorescence, right. Fluorescence. This also I explained, if the electron is flipping, goes from singlet state to triplet state. That singlet state means like the S is 0, so 2S plus 1 is 1 singlet state, triplet state S is 1, 2S plus 1 is 3, that is a triplet. Triplet state it goes, and that is called inter-system crossing, the system has been crossed. So, inter-system it crosses to from singlet state to triplet state, that is a, and then from there it comes to ground state, it is called phosphorescence. This happens in microsecond time scale. So, photon is emitted from triplet state, that is called phosphorescence. And yeah, and there are some non-radiative dissipative process that happens, we call it quenching. There is another terminology I will go and talk later on called FRET, we will talk it later, fluorescence resonance energy transfer, but at the moment let us stop there. So, absorption from S0 to S1 S2, emission happening in fluorescence S1 to S0, phosphorescence emission happening from T1 to S0, typically slower because of spin forbidden, right. If this is going, this was like this, it goes like this, it is allowed, but agar palti maar diya, I will not say it is not allowed, but spin forbidden, it takes time, and that is why it is delayed process, right. So, that is a inter-system crossing. So, energy dissipate without, it is also possible energy dissipate without giving any radiation, that is a phosphorescence. Is that okay? So, now Jablonski diagram is clear, this is extremely important diagram in fluorescence and phosphorescence, it should be hardwired in your brain. Which book do you want to read for this? How many of you want to make a career in fluorescence related work? In general, like in fluorescence itself is a very phenomenal spectroscopy. There is a book, if you want you can invest that money, Lakovich book, that you should buy and keep it. You do not think that after MSc fluorescence is going to leave you. If you are in science, whole your life you remember fluorescence. और कभी भी इस book की तुम्हें ज़रूरत पड़ेगी. If you are like a professor writing a manuscript and if you are asked you something tricky question and that you probably or your student are not able to explain, either you go to a more expert who is your colleague or go to even more expert which is book, right. So, you can read the Lakovich book. I recommend you to buy one copy for yourself. It is long term investment, right. It does not cost too much. एक मूवी और पॉपकॉर्न के बराबर है उसका प्राइस. It's just that you should buy a book. Now, coming back to the shift that we are talking about, the Stokes shift. We already discussed the basic concepts. So, when it goes from ground state to S1 S2 state, but when it is coming back it only comes from S1 state, बाकी energy loss. So, our delta E for absorption is higher, delta E for emission is lower. Therefore, wavelength is more, and that shift, the wavelength is more, that shift is called Stokes shift, because this phenomenal scientist reported this is called a Stokes shift. So, it is a, in which color it is shifting? Here is a blue, here is a red. So, always emission is red shifted, always emission is red shifted. So, excitation happens at a lower wavelength, emission happens at a higher wavelength. Ulta, absorption happens at a higher frequency and emission happens at a lower frequency, right. So, emitter at a longer wavelength or lower energy. Now, there is another important rule that I want to explain you, it is called CASA rule, states that fluorescence and phosphorescence emission in a molecule occurs predominantly from lowest excited electronic state, lowest excited electronic state, that means S1 or T1. How electron reaches to S1? By non-radiative process, that was internal conversion. How it reaches to T1? Similarly, intersystem crossing and then internal conversion it reaches to. So, that is a lowest excited state of given multiplicity, whether it is singlet or it is triplet, regardless of the which higher state it was excited. Even it was excited to S3 or S2, it always comes to S1 or T1 and from there it comes, right. So, when molecule absorbs light, it can go to higher state like S0 to S3. However, before emitting light, it does internal conversion and then internal conversion goes to different vibrational states, come to S1 and from there it comes to S0. So, say some molecules that exhibits fluorescence from higher excited state due to unusual their electronic structure or slow internal conversion, they are rare and often used for stabilizing the photophysical application. So, that is the one of the application of this. Now, another important point which I will explain again by going to the board called mirror rule. Let me explain you what is mirror rule. It will be here it is given, but let me go slowly and explain you. So, remember in excitation what we are doing? We are starting with S0, there are various vibrational state, अभी rotational state छोड़ देते हैं, we will just go to S1 and S2, right. This is suppose we are doing it, okay. So, S0, right, this is possible, maybe I should use another one, right, this is possible. Here also S2 may not that tall, so I do not want to go to S3, something like this, right. Okay. Let us draw the spectra for this absorption. S0 and something like S0 to S1, S0 to S2. This kind of absorption we can have, right. When it is coming back, what it should show when it is coming back? This is correct or not? Correct. When it is coming back, from where it comes? S1 to S0, S1 to S0. When it comes from S1 to S0, when it comes from S1 to S0, S0 may here comes or somewhere different vibrational state, right? Is that okay? Now, when we are saying mirror, so that means for this guy it is like this and like this, but for S2 will it be mirror? Understand my question? When it is going from S0 to S1 absorption, it is coming from S1 to S0 to different vibrational state, that is like a mirror, right. But when it is going from S0 to S2, will it come like same radiative process? No, always comes from S1 to S0. So, there is a whatever absorption that happens, S1 to S0 is mirror, but others are not that mirror. So, if you typically look at only S1 to S0 is S1 to S0 is a mirror, that is what the others are not happening. But in that it can go to different vibrational, S1 to S0 can go to different vibrational state, and from S1 it can come to different vibrational state of S0. So, there it is perfectly fine and it forms a mirror. Does it make sense? So, upon return to ground state, fluorophore can return to any ground state vibrational state, any ground state vibrational state. From S1 to it can be S0 V0, S0 V1, S0 V2, S0 V3. When it is going S0 V0 to S1 V0, V1, V2, V3, V4, right. So, this spectra typically shows mirror in the first transition level, beyond that it is not, because it goes in absorption excited. Conceptually this is clear, right. Right. So, if I just go back here in this diagram, if you look at which one will be showing mirror image and which will not show mirror image. Here the first red one will show the mirror image, that will not show the mirror image. Mirror rule is kind of quantitative, depends upon if there is no competitive like a quencher or something like that. But if quencher comes, pura game change ho jata hai, then it does not. So, typically all the case from S0 to S1 coming back S1 to S0 is quantitative. Right. Now, coming back to the how we detect it, conceptually this is fantastic. We looked at fluorescence is very important technique from all the way biochemistry, molecular biology, cell biology, neuroscience, developmental biology. If you want to see how my zebrafish is growing, like Srilaja does it, how zebrafish is growing, you mutate some gene in zebrafish, make it green fluorescent, and you can see how hatching is happening, how heart is beating, right, of zebrafish, and you can see it. It is just beauty of fluorescence. Developmental biology, आदमी का heart तो नहीं देख सकते, but at least we can see zebrafish heart, how it is beating and growing. It is a fantastic phenomena that is happening and can be seen using fluorescence, whatever biological science that you are doing, right. Now, how do you see it? What all it involves to absorb, to notice or determine it or to study it. So, first of all you need a fluorophore, right, and fluorophore that is without quencher. So, you need to have a fluorophore. Rest what you need? An instrument, a basic instrument you need. So, you need a light source. You remember what was the use for the first experiment that was done? You had a light source, you have a filter, you have a molecule, and you have again some filter and detector was eye. That is the basic things you need. So, here again you require an light source, an excitation filter, and dichroic filter, emission filter, detector, and sample holder. That is what you require. Just send the light and whatever light comes you detect it. That is the basic instrument that you need. So, high energy light source which can excite the molecule, you can have a UV lamp which excites the molecule, or laser if you want to be particular. Right. Light source UV light ka problem hai, there are too many frequencies that are contributing, but you want to be very specific, you require an intense laser source. As I said, fluorescence efficiency depends upon how many competitive pathway you have. So, sometimes you require a high energy for excitation and you can use for that laser source. If you are doing the experiment in cell, you require a laser light. Otherwise, for a typical biophysical experiment, a cleaner solution where your protein containing one tryptophan is in buffer, you can do with simple light, like a UV light you can do it. So, you require a UV light, a detector which can detect the fluorescence, and sample and then you have to cancel the whatever coming from the background like buffer or something. Your sample holder should not interfere with that. So, you can have a good cuvette where your sample is PET, that is a quartz cuvette, you can take it. And now, you have to have a sensitive detector where electrons are coming and getting like a photons are coming and getting detected. So, then the emitted light can be collected and analyzed by a detector, right. So, you require a detector. The intensity or wavelength you have to detect studying the fluorescence, and you can plot a spectra. So, this is a basic setup of an spectrophlorimeter, the instrument that measures the fluorescence spectra, that is called fluorimeter. What we had, the UV visible, a spectrophotometer, it is a fluorimeter which measures the fluorescence spectra, that is called a spectrophlorimeter. So, in your opinion, let us do the cost analysis of this, right. This is a basic instrument. Typically, for doing a biophysics experiment, how much money you want to invest in this? UV visible 10 lakhs. Now, giving you some idea to think about it. UV visible spectrometer that you are purchasing cost you 10 lakhs rupees. JASCO ka 10 lakhs mein you can get it. Okay. Now, for fluorimeter, how much money you want to invest and why? Will be cheaper, costly? Costly. Why it is costly? Detector thoda sensitive chahiye, that is the one thing. Light source thoda intense chahiye. Cuvette अच्छी quality के चाहिए. There should it should be nicely packed into a box, and then has to be computer where you can really plot this. So, all of these will cost you more than whatever UV visible spectrophotometer is. It is the basic one. Then if you go even beyond, you have to have a laser light, right. Laser will be costly. It is not the laser that you use a pointer, it is a nice laser you have to have. So, that depends upon from few lakhs to maybe few crores you have to invest in getting good fluorimeter, right. So, if you want to do cell experiment, you require even more intense light. So, fluorescence measurement, that is what we do, basically captures the how sample absorbs and emit the light and provide insight into composition of this molecular interaction. There are two kind of measurement we do. One is typically emission spectra. So, the light is emitted from S1 to S0, that is what we measure and plot against wavelength. Light is emitting, so what is emitting? Photon you can put it. What is the unit of emission? Nothing, arbitrary unit. So, y axis arbitrary unit, x axis you can have either wavelength or frequency, any of these you can have. So, if we are plotting the emission spectrum, then we have a wavelength versus arbitrary unit of photon that we measure. Detector scans the wavelength and detects the emitted photon, that generates the emission spectrum. But also important to do this experiment called excitation spectrum. So, that again it is a similar to absorption, but here we call it excitation spectrum. So, excitation, you shine the light to the fluorophore that excites it to ground, from ground state to excited state. First excited state, second excited, third excited, whatever, you plot it. Again it is a wavelength versus number of photon that is used. So, that is a excitation spectrum. So, why you need to do it? If you are a careful scientist, why you need to do it? Because your emission depends upon excitation, right. So, if you are not, like if you remember, let me draw a little bit. You see, our fluorescence is not that strong signal, it depends upon many competitive process, right. So, what we want to do is put the maximum light that we can. So, in that case what we have to do is you scan it something like this, your excitation spectrum, and choose the lambda max where your excitation is happening maximum, then you plot the emission spectrum. So, first in any case if you are doing the experiment, you have to do first excitation spectrum, then emission spectrum, that is the correct way of doing fluorescence. First you scan say from 200, so 300, right, and you found that your lambda max is coming here. Now what you choose? Say this is coming at 278, okay. Then you scan say from 300 to 400, emission comes at 340, that is the emission spectrum. Suppose I chose here, will this give me emission spectrum? Yes, it can give you, but intensity will be less, your signal detection or photon count will be less. So always the correct way of doing, first do the excitation spectrum followed by the emission spectrum, that is the correct way of doing fluorescence. So take your time, get the lambda max for excitation, then to emission. Now another two concepts I want to discuss. Up to here everything is okay?
It works as
That's a, you have to choose the mirror that can, essentially it is a simplistic diagram, it does not happen like this, that is simple. You have to have, like what is essentially, have to a mirror or a optics which sends you light to the sample, and when it comes it sends back to the detector, that is what. So you can choose it depending upon, that is what alignment and optics you have to do, right. So it is not that simple, it is a simplistic diagram that is done, it is not a simple mirror arrangement that can you do with only one mirror. Okay. Now two important concepts we want to discuss. We already discussed in bits of this. Fluorescence depends upon the molecule itself and what is the competitive process around it, right. Competitive processes were what? Internal conversion, intersystem crossing, quenching, non-radiative process. There are too many that are there, right. So efficiency of a fluorophore is determined by what is the radiative process, what is the non-radiative process, and that efficiency is called quantum yield. Quantum yield. So quantum yield is the ratio of number of photons that is emitted by number of photons that was absorbed, right. So this is the that is what you have, the quantum yield. So quantum yield of different fluorophore can vary. Some fluorophore can give you very high quantum yield, some can have very low. Now let us look at the three amino acids that we discussed in absorption. What were those three? Tryptophan, tyrosine, all these three were absorbing in UV light, but they do not have same efficiency when they emit. Phenylalanine, it absorbs, right, at 260 or something, 265 or something. When it has to emit, it does not emit, just takes the photon and be there. Tryptophan is the most profound one, it emits, so its quantum yield is high. Then comes tyrosine, it also emits, not that significantly. So if you look at in protein, out of these three, two are used for fluorescence. The favorite one is because of its high quantum yield. Because of its high quantum yield, it gives you substantial emission. That is why its quantum yield is high and it is used for doing the fluorescence experiment. Is that okay? So that is a quantum yield. Now the another thing is lifetime of a fluorophore. Lifetime, the lifetime of excited state, how much time it stays in excited state. Why it is important? Because this gives you some property of a fluorophore. So how much time it stays in excited state, that prior it coming back to the ground state, that is called lifetime of a fluorophore. So quantum yield Q can depends upon the total time tau plus rate of non-radiative plus radiative. So K, that big lambda is emission rate of fluorophore, and KNR is rate of non-radiative fluorophore. So lifetime of an excited state defined by the average time that molecule spend in excited state prior to returning to the ground state. Two concepts, one is quantum yield, efficiency of a fluorophore, second is how much time it spends. Right. Immediately if it returns, then it will not report something, but it is stage, because fluorophore is a probe, probing something. So, if it is stage, it has a lifetime, it reports something, and then we can use this as a reporter to understand the biological phenomena or conformational change or something happening. The lifetime is the time that it spends in an excited state. These two concepts or these two parameters are extremely important for a fluorophore to be there in excited state, coming back to ground state, the lifetime and the quantum yield. So, just I will take 1 minute why lifetime is important and what it can report. डिले तो पता चल रहा है कि उधर रुक गया तो डिलेड है, राइट तो उससे क्या फायदा हुआ, व्हाट बेनिफिट आर वी गेटिंग, सॉल्वेंट इफेक्ट, सराउंडिंग, व्हाट इज द सराउंडिंग environment. So, suppose just for a protein sake, a tryptophan like this or tryptophan like this, will they have different property? Yes, this is doing like this, right, and this is not doing like this. So, if I measure something called lifetime of a fluorophore, which is free movement versus restricted movement, a simple experiment of fluorescence lifetime measurement is giving me idea whether my fluorophore is moving like this or like this. So, the lifetime measurement is giving you idea of the structure तो नहीं है भाई, ये क्या हो रहा है, motion dynamics, it is giving the idea about the dynamics. My tryptophan is moving like this or my tryptophan is moving like halka sa, right. It is also possible that this whole molecule goes like this and my tryptophan is sitting somewhere, right, it goes like this. So, it gives you idea about local motion, it gives you idea about whole molecule tumbling. So, lifetime is extremely sensitive experiment to understand the molecular tumbling, the local motion, the dynamics. Suppose my tryptophan is sitting here and it does motion like this, conformational switching, this can give you idea about that. Quantum yield tells the efficiency and the lifetime gives you the dynamics property. So, these are the two concepts that one can use it. So, advantage of fluorescence, it is a highly sensitive technique, right. You see how profound things it is giving you local environment information, it is giving you dynamics, simultaneously it is giving you dynamics, and how much you invest in getting this information, it is a sensitive technique. Only 10 micromolar of protein you require just for doing a fluorescence 10 micromolar. With investment of 10 micromolar of protein with single tryptophan, you can get the phenomena of molecular tumbling, the local motion, the environment and all those. So, it is a highly sensitive technique and also selective. So, you can use for various things, right. So, it is a 1000 times more sensitive than absorption spectroscopy method, detect very low concentration, because we have a reporter which is very specific, right. It is a Dhurandhar, we have sent it in the protein and can report it selectively. It is a non-destructive because we are not destroying the sample. For getting this information, we are not destroying, we can, after doing the experiment, we can take it out, do something else. It is a non-destructive, and like if we are doing mass spec जहाँ हम तोड़ते हैं bond को, here you do not do anything, you do not do any harm to, it is a non-destructive process, highly selective. So, only emission is coming from that particular tryptophan where, or that particular fluorophore whatever we have tagged, and it is a very informative. So, fluorescence can provide the quantitative, qualitative, includes the concentration, trace detection, many things one can do. It is a versatile technique that I am repeating it many times. So, it can, we can do various experiments in various field including biophysics, right. What is limitation? Everything is not good, there are some limitations that come. First and most important limitation, that all molecules are not fluorophore, one is selective molecule. So, you have to find a fluorophore, right kind of fluorophore. So, see, there is a business opportunity for yourself. Synthesizing chemistry guys or biology guys can combine their like intelligence to bring a nice fluorophore molecule that has a high quantum yield. Still there are many, but still you require many more, that is highly sensitive and has high quantum yield. So, that is a molecule is like there are only limited fluorophore that one can use it. The another problem that it is autofluorescence, naturally emits fluorescence, and indigenous fluorophore can interfere by analysis of targeted molecule. Many times you do not know what is happening if you have a fluorophore, you are not getting whatever spectra, because of autofluorescence is a possibility of your molecule. Now, it is very sensitive to environment. So, if you have a something quencher sitting there, you do not know my protein is not fluorescing because you do not know it has been quenched. So, environmental factors are there, it can be temperature, it can be pH, it can be solvent, all of these affects the fluorescence. Now, many times you do not have a fluorophore in the protein or a biomolecule, then you add it from extra. How do you add it? Suppose I have a protein, I want to attach a fluorophore. How do you attach it? Fluorophore has to be attached, it should not move here and there. Infusion, gene fusion. So, you are not attaching a small fluorophore, right? You are attaching a big one. GFP is a big protein. Say my protein is of 10 kDa and GFP is of 40 kDa, right? ये तो ऐसा हो गया ना, इतना छोटा नीडल उसमें इतना बड़ा है thread. You have to think of a smart way that you can do, that is a way, but you think of a smart way. In 100 amino acid you cannot add 600 amino acid and say that it is all fine. So, what else? And my protein does not have any tyrosine tryptophan, but you want to do this, you want to use this protein. How you can use it? You add an extrinsic fluorophore, you can add it to a particular amino acid, then it will be more selective, right. You can attach to a particular amino acid. Those amino acids where you can attach are, you have to do a covalent modification. Where is a possibility in protein you can do covalent modification? Serine, serine में OH है. The best one is cysteine. Disulfide bond can be easily made. So, many of these tags are attached to. Some non-selective attaching you can also do. This guy lysine has a nice side chain. What is the side chain of lysine? There many molecule can be attached, but the best one, a selective one, cysteine. So, you can have the fluorescence attachment probe, you attach a fluorophore in the molecule and you can use that for reporting it, but you have to design this experiment very, very carefully. If your protein does not have a cysteine at all, you have to create a cysteine. कैसे करोगे? Point mutation, right? You can mutate it, create a cysteine mutant, single cysteine mutant, attach a fluorophore, this fluorophore is reporter for you, so one can do it. Another limitation is limited penetration, so less suitable for sample with a greater thickness, because fluorophore has to go and report it, and the infrared can penetrate, fluorescence cannot penetrate that much. The another problem is photo bleaching, right. So, continuous exposure of, if you just shine the light and take out the fluorescence, it is okay, but if you continuously keep shining the light, it is not okay, that is photo bleaching. But that is used in some of the experiment in fluorescence. You know what is this experiment? Called FRAP, fluorescence recovery after photo bleaching. अगर तुम्हारा molecule interact कर रहा है, so you shine the light, take out the light and let them relax and come back. You see dark and after some time it is light. So, you see in cell how the motion is happening, that is called FRAP. So, photo bleaching is a problem, but you can use that problem for advantage. Now, quenching and interactions, that is also reduces your fluorophore, and complexity, of course, I keep insisting एक tryptophan बहुत अच्छा, 2 tryptophan manageable, we can mutate one. If there are too many tryptophan, it is not good. It creates a problem for you, right. So, now last thing, if this technique is so profound where we can use it, already I have said analytical chemistry, right. You can detect the low concentration of substance in nanomolar or picomolar range, it is a very, very sensitive technique. Reaction monitoring, you can use a fluorophore tag and look at how reaction is proceeding, it is a continuous flow, you can do reaction monitoring. Identification, you can identify a molecule, differentiate the compound with a unique fluorophore. That is a like some chemistry, biochemistry application. You can have a biological or biomedical, right, fluorescence dye to measure a nucleic acid concentration, you know, even the cancer dyes are used. Suppose you are working on a cancer drug and you want to see whether cancer drug is binding or not. Name a cancer drug. No, the DNA binding cancer drug, let us be specific. Doxorubicin सबने सुना है ना, doxorubicin, it binds to DNA. So, when it binds to DNA, its fluorescence property changes and you can use that for looking at if DNA is interacting, what kind of DNA is interacting, if it is intercalating or not, breaking the DNA or not. So, those kind of experiments one can do it. Protein study, of course, protein study it is used very much, intrinsic as well as extrinsic fluorophore. You can image it, look inside the cell. As he was saying, you require your protein with GFP tag and now you can transfect inside the cell, and wherever your protein is going, this guy is showing the path, right. You can see how your GFP is fluorescing and going where, you can look, the cellular imaging one can do. Immunofluorescence, right. So, you can detect the fluorescently labeled antibody, if antibody is interacting with antigen or wherever it is going, immunofluorescence can be used. Diagnostic, cancer detection. Just for an example, when a doctor removes the tumor, how do it knows that where is the tumor? ग्लूकोज को लेबल करेंगे तो उससे क्या होगा? द ग्लूकोज. अगर हम कोई फ्लोरेसेंट डाई वहाँ भेज दें तो you can see that it will selectively illuminate. I give you an example, again it is a very profound. Cancer cell express something very high called folate receptor, right? Folate receptor. If I send a molecule which is attached with folate and fluorescent molecule, this will be uptaken by that folate receptor and it illuminates the tumor. Now, doctor can zoom it there and remove the tumor. So, in not even in detection only, but operation can also be helped. These kind of things people are already doing. Pathogen detection, it can detect virus, bacteria, other clinical sample, and a biosensor monitor like a glucose monitor, pH monitor and all those in real time one can use this fluorescence monitor. You can also use that environmental monitoring, pollutant concentration one can measure, the water quality testing can be measured, maybe pharmaceutical and drug development, of course, drug screening. When you develop a drug, from how much molecule, how many molecule you start. Suppose I want to develop a drug for Parkinson's disease. I know which protein is my target, but I do not know which molecule will work. It is a 10 years of search to get one molecule. Now, here fluorescence can help us. Why? It is a very sensitive technique. So, I have to label protein with some my interest of protein with some fluorophore and pass the molecule one by one. जो change किया वो binder है, जो change नहीं किया वो non binder. So, this high throughput screening of a drug molecule, potential drug molecule can be done using fluorescence spectroscopy. And of course, the tryptophan that we are discussing reports about the tertiary structure of a protein. So, the tertiary structure of protein integrity can be measured using the fluorescence spectroscopy. So, if you like notice it clearly, it has lots of technique. I would say we will continue this another topic in coming lectures. We will stop it here and I would like to take any question if you have.